So, hello and namaste to everyone. This is Name-Shalu Singh from Department of Physiotherapy and Diagnostics in Jayoti Vidyapeeth Women's University. So, for today's lecture, we'll be talking about protein buffers. So, in order to know what are protein buffers, let's revise what we have already studied in our last lectures. So, in our last lectures, we came across what are acids, then we came across what are bases. So acids are the substances that are capable of donating protons, and bases are those substances which are capable of accepting protons. So and we also went through examples of acids and bases. We got to know about weak acids and strong acids, and then we talked about a dissociation constant and a pKa value, and we also talked about acidity of solution, and the most important thing that we came across was Henderson-Hasselbalch equation. So in next lecture we talked about buffers and what are the different types of buffers, how they act, what are the factors that affect pH, what is buffering capacity, and what are the applications of these buffers. Okay, so later we talked about what are if we we talked about what are the buffers of body fluid. So our body has various mechanisms for regulating its pH. So as we have already known that a pH of a blood plasma is 7.4, that is in the ranges of 7.38 to 7.42. So any decrease in pH below 7.38 may lead to acidosis, and any condition above 7.42 may lead to conditions of alkalosis. So our body then has several mechanisms or a defensive mechanisms in order to regulate its pH. Okay, so first, a line of defense is your blood buffers, which we are currently studying, such as your bicarbonate buffer, phosphate buffer, and your protein buffers. Second line of defense is your respiratory regulation of pH, and then third line of defense is your renal regulation of pH, which both of the second line of third line of defense will come across in our future lectures. So for today's lecture, let's go upon to let's see upon what are bicarbonate buffers. So in bicarbonate buffers, I've already told you that these are one of the most important buffers, and a bicarbonate buffer has a bicarbonate carbonic acid system, that is NaCO3 and H2CO3 system, and it has a 65% of buffering capacity in plasma, so which is a lot, like 65% is a lot, and 45% buffering capacity in our whole body. So now bicarbonate buffer, they'll have a two components, metabolic component and your respiratory component. And one of the most important thing that you need to know in bicarbonate buffers is that the ratio of HCO3 minus to H2CO3 minus bicarbonate to carbonic acid at pH 7.4 is 20 under normal conditions. So this can be asked in any of your competitive exams, so you definitely need to know about that. Then next we read about phosphate buffers. So these are also intracellular buffers, and the concentration is, since the concentration is in plasma is very low, so they are helpful definitely, but not more than your bicarbonate buffers. So pKa value is basically 6.8. So now, since after phosphate buffer, in also phosphate buffer, we read about PBS solution, phosphate buffer saline solution, which is of great help in your researches and all. So today we'll be talking about your protein buffer system. So protein buffer, firstly, one of the most important was your phosphate buffer. Secondly, your phosphate buffer system. Protein buffer is also one of the most important uh, buffering system, lower than your bicarbonate buffer, but since it is in your body, so it is important. So buffering capacity of protein, it depends upon your pKa value, that is your pH of your dissociation constant value of ionizable side chain. So the most effective group is your histidine imidazole group. So you should always remember this thing. It is the most important point of protein buffer system, that the most effective group is your histidine. Histidine is your amino acid. So histidine imidazole group with pKa value of 6.1. Now, the role of hemoglobin buffer is considered along with respiratory regulation of pH. So, a protein buffer system, it is important, but its main important role is there during your respiratory regulation of pH. Now, for protein buffers, we'll be going across few further stuff. So, which I'll be telling you right now. So, further, we'll see how protein buffers act. So, protein buffers, they behave as buffer in both plasma and cells, which I've already told you. So, hemoglobin is by far the most important protein buffer and the most important intracellular buffer, so which is ICF. So the most plentiful buffer, it is the most plentiful buffer of the body. So the proteins are excellent buffers because they contain both acids and bases basic groups, and they can give up or take up hydrogen ions. And proteins are extremely abundant in cell. So the most important proteins that we have is in our cells and in our blood system is your albumin and globulin. And when you study biochemistry in detail, you'll get to know that albumin and globulin, they act as carriers for most of the components within our body. So protein, basically these proteins, they act as carriers, or they hold certain substances, or they may transport a substance from one location to another. So protein buffers, they're very important, and they are they are extremely abundant in our cells. So the most limited number of proteins the more limited number of proteins in plasma reinforce the bicarbonate system in your extracellular fluid. So the hemoglobin buffer, it buffers hydrogen ions from metabolically produced carbon dioxide in the plasma only. So if you read about the structure of hemoglobin, it can get attached with 4 of the oxygen ions. So if there is one oxygen ion attached to the hemoglobin, it will increase its affinity for other oxygen ions to be attached. So one oxygen ion, there is increase in affinity for the attachment of other oxygen ion. If second oxygen ion, there is even more affinity for uh, other oxygen ions to get attached. So there is attachment of third oxygen ion. So further, when there be a third oxygen ion attached, there will be affinity for fourth oxygen ion attachment. And since there are only 4 spaces for oxygen to be attached, so at 4 attachment of oxygen ion, it gets stable and no more oxygen ions can get attached. So this is the basically structure in the structure of hemoglobin. So one hemoglobin, it can carry 4 oxygen molecules. Now later we'll see that hydrogen generated at the tissue level from dissociation of bicarbonate produced by the addition of carbon dioxide and bound to hydrogen ions to hemoglobin. It does not contribute to the acidity of blood. Right now, the hydrogen hemoglobin, this complex is formed, and then it picks up oxygen from the lungs, and the hemoglobin, which has higher affinity for oxygen, releases hydrogen and picks up oxygen. Right So it is a liberated it hydrogen ions from water. It combines with HCO3 minus, and this HCO3 minus, it will if we write it in equations, this HCO3 minus will give H2CO3, and it will get further give CO2, which is exhaled, right, through our lungs. Now, uh, since the venous blood is only slightly more acidic than arterial blood because of the tremendous buffering capacity of hemoglobin, even in spite of large volume of hydrogen generating carbon dioxide carried in venous blood. So protein can act as buffer for both acids and for both bases and acids. So protein buffer system works instantaneously, making it more powerful in the body. And 75% of the body's buffer capacity is controlled by your protein. So protein is also one of the most important buffers within our body. First one is your First one is your bicarbonate. Second, after bicarbonate, protein is also very important, and phosphate is least important because of the less concentration of phosphates present in our body. So, bicarbonate and phosphate buffer systems, they require several hours to be effective, and protein buffers, they can act instantaneously because they are present in our cells and tissues. So proteins are very large and complex molecules in comparison to size and complexities of acids and bases. So basically, in a manner, we can say that acids, when you read about the structure of proteins, so proteins are very complex molecules. They are present in primary, secondary, quaternary, and tertiary structures, basically. So, and mostly proteins which in our body, they are present in tertiary and quaternary structures, such as hemoglobin and collagens. They are mostly tertiary and quaternary structures present. So proteins, they are surrounded by multitude of negative charges on the outside and positive charges in the inside or in the surfaces of the molecule. Now, next, the hydrogen ions are attracted because outside is negative and the hydrogen ions are positive. So opposite charges, they'll attract each other. So these the negative ions present on outside, they will attract your hydrogen ions. So the hydrogen ions are attracted to and held from chemical interactions by the negative charges. Now, the OH minus ions, which are also present. So, because outside is negative, so outside the portion of protein, it will always repel. But in the cervices or within the interior of proteins, positive charges are present. Those positive charges, they will attract those OH minus ions. So OH minus ions, which are the basis of alkalosis, they are attracted by the positive charges which are present in the cervices of your protein. Now, so a protein ion, as I told you, now they can uh, affect both proteins, acids and bases. So this is how, because proteins, they are having negative and positive both charges, so they can, according to the convenience, they can affect both your acids, that is your positive charges, or your bases, your negative charges. Fine. So this will be it for your protein buffers. So now in later chapters, or we'll be talking about buffer capacities. Firstly, we can always today we can also learn about what is a relative capacity of buffer system. So in body, 52% buffer capacity is in tissue cells and 6% is in your RBCs, and rest 43% is by extracellular buffers, and in plasma and extracellular space, and about 40% buffering action is by bicarbonate system, and 1% is by proteins, and 1% is by your phosphate buffer system. So buffers act quickly, but not permanently. So how? Buffer, they can respond immediately to addition of acid or bases, and they do not serve to eliminate the acid from the body, and they're also unable to replenish the alkali reserve of the body. So for the final elimination of acids, the respiratory and renal regulators, they are very essential. So in later classes, we'll be reading about, we'll be studying about respiratory and regulation of pH and renal regulation of pH. Now, I'll end it up here, and we'll meet in next class. Until then, do read about the whole acid-base balance and acid-base regulation, and don't read about the buffers and biological regulation of pH. And I'll meet you in next class. Until then, if you have any doubts, do contact me, and I'll clear your doubts. And until then, thank you so much and take care.